Hello everyone, welcome to Vidyalakshmi YouTube channel. Today we are going to see the chapter Heredity, and here generally children think that Heredity is the difficult chapter, but believe me, Heredity is the easiest and very interesting chapter in biology part. So here the term heredity defines the transmission of the characters, genetic characters from the parent to the offspring. So in this chapter we are going to study about different terms related to heredity, and there was a scientist, well-known scientist, who is called as the father of genetics, that is none other than G.J. Mendel. We are going to study about his experimental details. And here, before beginning to the chapter, this session is divided into two sections. One is the introduction to genetics. So here we will be dealing with all the basic concepts related to genetics, and then we are going to see about Georg Johann Mendel and the experimental detail. So this scientist, Georg Johann Mendel, is the person who is called as the father of genetics, as we all know. So every cell have a centrally dense material called as the nucleus. So this nucleus will have the thread-like structures called as chromosomes, and within the chromosome we have the nuclear material called as DNA, and within the DNA we have the functional portion called as the gene. So what is the relationship between all these terms here means chromosomes are the structures which contains the DNA, and DNA in turn contains the genetic information in the form of the structural sequence called as the gene. These three characters play a very important role in the transmission of the characters from one generation to the next generation. So if you just look at chromosomes, chromosomes are nothing but long thread-like structures which are present in the nucleus of a cell. The thread-like structures, especially during the cell division, the thread-like structures which are present in the nucleus of a cell is called as the chromosomes. So this chromosomes contains the genetic information in the form of the genes. So here you have the genetic material present called as the DNA. So this DNA is the chemical which carries the traits in the coded form. So this DNA is nothing but the genetic material called as deoxyribonucleic acid. So within the chromosomes we have this genetic material present, and this DNA is a long sequence. In this long sequence, the entire sequence is not going to be the useful one. So in this DNA sequence, we have the functional portion called as the gene. So here gene is the part of a chromosome which specifies the biological function. So every character in our body, right from the hair color or our pupil color or the entire characters or the body design of a person depends on this particular gene only. So every gene codes for a specific proteins inside our body. So here if you see, every DNA contains a structural element called as alleles. Within chromosome we have DNA. Within DNA we have the gene structures, and within genes we have the structures called as alleles. So these alleles will be represented by two letters. Either it will be in the capital letter or it will be represented in the small letter. All the dominant alleles will be expressed in the capital letter, and all the recessive alleles will be expressed in the small letter. So these alleles are responsible for the determination of the character. Any characters are determined by the combination of all these four structures. So as I told you before, nucleus contains the chromosome, chromosome contains the DNA, and within DNA we have the functional portion called as the gene, and within gene, every gene will have a specific alleles present inside them, and they are responsible for the characters which we get. So different versions of a allele or different versions of a gene is called as the allele. Alleles are described as either dominant or recessive depending on their associated traits. So here the word trait meaning is character. So what character it determines, based on that we are dividing the alleles into dominant and then the recessive form. A dominant allele produces a dominant phenotype in individuals who have copy of the allele and which is coming from just one parent. So if you altogether see children, these alleles are nothing but the specific structures which are the part of the gene. So any individual if you take, that is which is undergoing the sexual mode of reproduction, so we will be getting, suppose I have a skin color. So this skin color is a combination of two allele. So one allele will come from the male parent and another allele will come from the female parent. So we have a fusion of both the alleles, and because of that we have the determination of the character. So based on which character is getting expressed on how the combination of the alleles is determined, we are dividing the alleles into two types, that is nothing but dominant allele and the recessive allele. So on the combination of two alleles, the dominant allele requires the presence of only one. If one dominant allele is present within the gamete, it is enough for their expression. But if you look at the recessive allele, for the expression of that particular recessive character, we need the combination of both the alleles to be recessive in nature. In case if I am telling the plant is, in case if you are taking the example of the stem height of the plant, if you can consider capital T as the allele which is responsible for the dominant character and small t as the allele which is responsible for the recessive character. So here only capital T is enough for the expression of the dominant nature. But if you want the characters to be expressed in the recessive form, it requires the presence of both the allele structures. So here, if you take as the example, in flowers, no, we have two different kinds of flowers. One is the violet color flower and other one is the white color flower. If this violet color flower, okay, which is dominant in nature, is determined by the allele capital B, and the white color flower which is recessive in nature is determined by the letter small b, then for the expression of the dominant character, either the alleles should be the same, that is capital B, which we are calling it as homozygous, or even if it is capital B, small b, that means the presence of one dominant allele alone is there, then also the flowers will have the violet color. That is the power of the dominant allele. So every gene will have two alleles present within them, and that gene is responsible for the particular character. So in this example, if I am taking flower color as one particular character, so flower color violet is the dominant one and white is the recessive one. For the expression of this dominant nature, we have the dominant allele present called as capital B. For the expression of the recessive nature, we require the allele called as small b. Now, why we have to call capital B as dominant in the sense, for the expression of this particular character, the presence of one allele is sufficient. For example, capital B, small b is there, only one capital B is present. Then also, the plant will execute the particular character. Expression of the recessive allele, unless and until both the alleles are recessive, that means both are small b, then only the particular recessive character will be expressed. So if you can revise again, a dominant allele produces a dominant phenotype. Phenotype is nothing but morphological appearance or the physical appearance of a particular character. So who have only one copy of the allele which comes from any one of the parent. A recessive allele, to produce a recessive phenotype, the individuals must get what? Two copies, that is one from each parent. So if it is present together, if both the alleles are recessive, then only the particular recessive character will be expressed. So we have two types of alleles called as what? Dominant allele and then recessive allele. Again, based on the composition of the alleles, we can categorize the alleles as homozygous and heterozygous. All these terms are very important when you work out the Mendel's experiment. So here we have seen about chromosomes. Chromosome in turn contains the DNA. DNA in turn contains the gene, and within gene we are studying about the alleles. So as I told, alleles are responsible for the expression of the character. So here, based on their expression, we have previously categorized them as dominant and recessive allele. So based on their structure, here we are categorizing them as homozygous and heterozygous alleles. The term homo means same. The term homo means same. And the term hetero means different. So these alleles are present on the structure of the chromosomes. Chromosomes are called as the gene carriers. They are the substances which are carrying the gene from the parent to the offspring. So if chromosomes are carrying genes, obviously the alleles are present on the surface of the chromosome. So here if you take this particular chromosome, number 1 here, the chromosome is having capital B on one sister chromatid and another capital B present on the other chromatid. If you take chromosome 2, small b allele is present on one chromatid and another small b is present on the other chromatid. So the chromosome number 1 have capital B allele present on their sister chromatids. Chromosome number 2 have only small b alleles present on both of their chromatids. So from this, since the alleles are same, I am giving the term homozygous. Homozygous allele represent the same type of alleles present on the structure of the chromosome. Here the opposite term heterozygous. If you look at chromosome number 3, here one chromatid is having capital B, that is the dominant allele. Other one is having small b, that is the recessive allele. So a particular chromosome carries two different alleles. A single chromosome is carrying two different alleles on their chromatid structure. So such chromosomes are said to have heterozygous allele. So if the alleles are same, it is called as homozygous. If the alleles are different, it is called as heterozygous. Here look at the chromosome number 4. Here small b is present on one chromatid and the capital B is present on the another chromatid. So if you have same allele, it is called as homozygous. If you have different allele, it is called as heterozygous. So here we are entering into the chapter. What is called as genetics? Genetics is the field of science, like how we study botany. So botany means we will remember the term. So botany is a study about the plants. Zoology is a study about the animals. Like that, the word genetics is a field of science where we study about heredity and the variation. Heredity is nothing but transmission of the characters or the traits from one generation to next generation. That means from the parent to the offspring. What about variation? Variation is nothing but the morphological differences or the genetic differences that exist among the population. So let us see them in depth. Heredity, here the transmission of characters or traits from one generation to next generation is called as heredity. Transmission is nothing but passing. The passing of the genetic contents from one generation to next generation is called as what? Heredity. For example, if you look at yourself in a mirror, you will have your body will have the combination of characters from both your father and the mother. That is nothing but heredity. The characters are passing from one generation, that is from our parents, to the next generation, that is none other than us. So this kind of transformation is called as heredity. If you look at this picture, you can able to see the similarities that is existing among the offsprings or the progenies, okay, from the parents. So they can be the transmission of the characters such as the highest pupil color. Highest pupil color, it can be black, brown or even bluish in nature, that comes from our parents. And here if you see the ears, some people will have the attached ear lobes. Some people will have the free ear lobes. That is also coming from our parents. If you look at your own height, the height, okay, to which we are growing, no, that is also the character which we are getting from our parents. And some of us will have the plain hair pattern, some of us will have the curly hair pattern. Everything comes from our parents. So heredity is the one which is dealing us with all the things. Not only in human beings, but also in all the animal species, okay, like fishes and all, if you take, no. So if there is a cross fertilization between two different species having two different characters, this is a simple cartoon work which tells us about the heredity. For example, the fish number 1 have the characters, okay, which are given as red color. Here the fish number 2 have some characters which are given as the blue color. Now when you see the progeny will have the combination of both the characters. That is, the characters will come from both the parents, from number 1 and from number 2. So the hybrid species or the progeny will always have the combination of both the characters. In plants also, if you see, no, this is just the flower image which is given here. If you look at this particular image, no, you have the combination of two colors here, the rose color and then the white color. So you have the combined or the hybrid content of both the colors. That is nothing but you can tell by yourself, no. So here this flower is a hybrid plant arised from two different species. So the plant 1 would have been producing the pink color flower or the rose color flower. Plant 2 would have been producing the white color flower. So when you make them to cross-pollinate by any kind of the sexual methods, here the new hybrid obtained have the combination of characters. How the combination of characters obtained? It is because of the concept called as what? Heredity. So heredity in the sense, no, you have the transmission of the characters from one generation to the next generation. I think you are clear with that. Now the next term. So as I told you, genetics deals with heredity and then the variation. So here the term variation. What is meant by the term variation? The differences in the characters or the traits between the parents and the offspring. So what are all the changes which can be seen between the parent organism and the next generation offspring organism is called as variation. For example, if you look at this particular image, we have different categories of people, different age categories of people are there. So if you see and compare yourself, for example, if you are inside the classroom, maybe around 20 to 25 children, and if you check yourself with all the other members in your class, definitely you are not going to be the same. Definitely our morphological structure will be different. Even though human beings, if you take as an example, our body design remains to be the same, the morphological difference exists. That is nothing but called as what? Variation. Not only in human beings, as we have seen many examples earlier, in butterflies, if you see the colors of the wings, they have different colored wings. And that is all arising the concept called as what? Variation. Human beings, skin color. We have different skin tone, right? Some of us will be having a dark skin tone. Some of us will be having a medium skin tone. Some of us will have a fair skin tone. Everything is called as the variation. Even in birds, if you see, no, the shape of the beak, it is the example for the variation. The beak pattern of each and every bird is different, and that is the example for variation. Again, if you look at the flowers, we have different colors of flowers. See, everything belongs to same species, right? Then also you have some changes among them. Some differences are present among them. That is called as the variation. So overall, genetics is the subject where we are studying about heredity and then variation, which I have explained you along with the example. I think you should be clarified with the concept of heredity and the variation. Now, this kind of variation exists in both asexual reproduction as well as the sexual mode of reproduction. In this ASR reproduction, if you take only one parent is involved, you can raise question, ma'am, only one parent is involved in ASR reproduction. Even though what is the possibility for the variation to take place means, in ASR reproduction, we have a process called as DNA copying. We have a process called as DNA copying or replication. It is nothing but replication means making a copy. So the parent organism will have one particular DNA. This is one particular DNA with a helical structure. Now this DNA will undergo the process called as replication. So DNA is said to have double helical structure. Helical in the shape, you will have the helical pattern like this. They have double helical structure. During this replication, what will happen is, using the enzyme called as helicase, this DNA double-stranded structure will unwind. That means, no, each strand will have a bonding between them that will break, and then they will become a separate copy. So this DNA is divided into two strands, and from this two strands you will have the polymerase enzyme which is capable of producing one more strand. So one DNA will give rise to two DNA copies, and this DNA copies will be equally distributed to the progenies, so before the cytokinesis process. During this replication process, how variation exists means, this replication process have the role of various enzymes in it. And when these enzymes are doing the replication and proofreading mechanisms, there is a possibility for the minor inaccuracy to exist. This minor inaccuracy lead to the variation in the species, even though it is reproducing by the asexual mode of reproduction. There also you have the probability of the variation taking place. And here, if you take the SR mode of reproduction, as we all know, here you have the combination of two parental characters. The egg is coming from the mother, or egg or ova obtaining from the mother is fusing with the sperm which is coming from the father. So when you have a fusion, the egg is carrying the parental characters from the mother and the sperm is carrying the parental characters from the father. So when you have the combination, there is a possibility for the recombination of the genes. There is a possibility for the recombination of gene, and as a result, the variation will exist in the next generation or the progenies. So these are all the basic concepts. Now before going to the experimental part, we have to be clarified with the concept of genotype and then phenotype. Without clarifying with these terms, understanding Mendel's experiment is very, very tough. So here the term genotype. The genotype is an organism's genetic information, the genetic makeup of the organism, or the genetic information about the particular organism is called as the genotype. For example, we have seen the allele composition. So if it is same means it is called as homozygous. If the alleles are different means it is called as heterozygous. So here, in case I am taking the flower color as an example. On looking at the flower, I can tell the color. So it is violet color or it is white color. But how can we predict the genetic composition of the violet color or white color flower? It is not possible. So this genotype will give us the information, that is genetic information about the particular character of an organism. The opposite one is phenotype. The phenotype is the set of observable physical traits. So we used to use the keyword observable. Observable means without using any microscope or without using any kinds of aids or experiments, just looking at the plant species or animal species, if we can able to predict some trait, that is called as the phenotype. For example, look at here. Here on looking at the flower, I can tell it is a purple color flower. The second is also the purple color flower. The third is the white color flower. This is called as phenotype. When I am telling you the physical appearance of the species, it is called as the phenotype. But when we are giving the genetic composition, so in case if capital B is responsible, capital B is the allele responsible for the dominant color called as purple, and here small b is responsible for the recessive color called as the white. So here the first plant purple is given the genotype capital B, capital B. So if it is same, it is called as homozygous, right? So this capital B, it is homozygous and dominant in nature because dominant character is purple. Now look at the second flower. The second flower is also purple in color. But if you see the genetic composition, it is capital B, small b, heterozygous, dominant nature. So here there is a difference between these two violet color flowers. The first flower is having the genotype capital B, capital B. The second flower is having the genotype capital B, small b. Then also why violet color expressed? As we have discussed earlier, capital B is the dominant allele and presence of one allele is enough for their expression. So capital B is also violet, capital B, small b is also violet. But what is the difference? Capital B is the homozygous alleles, and here capital B, small b is an example for heterozygous allele. And last one, violet color flower, if you take, as we all know, it is a recessive character. What is recessive character nature? Both the alleles should be recessive. So here this white color flower have small b, homozygous recessive. So on looking at the flower or on looking at the organism, if you are able to observe any character, it is called as the phenotype. But only when the genetic information is given, you can able to extract the information about the genotype of an organism. And all this variation has led to the evolution of the species. All this variation has not taken within a fraction of second or within a minutes or within an hours. It took millions of years to get accumulated in each and every generation. And now if you take human being as an example, we are Homo sapiens. We are the examples for the evolutionized species. When you compare our ancestors, our entire body design, pattern, the way of living, everything has been changed. This evolution is nothing but the complete accumulation of variation over several generations. So here we are going to see about the great person called as Gregor Johann Mendel, who is called as the man of science and man of the God. Gregor Johann Mendel is the person who is called as the father of genetics. He was the person who worked on the concept of inheritance of characters. Everybody is telling characters are getting or traits are getting transferred from one generation to the next generation. But what is the proof for that? Where is the experiment for that? For all this question, only one person who started this experimentation on inheritance of characters is none other than G.J. Mendel. The experiments he conducted is called as plant hybridization. Why it is called as hybridization means it involved the cross-pollination and self-pollination of two different genus, okay, which belongs to the same species. And here the plant sample which he has selected for his experiment is Pisum sativum. The common name of this plant is garden pea plant and the scientific name is Pisum sativum. There are some reasons why he has chosen this plant. So here the flowers are bisexual in nature. So the flowers will be bearing both male reproductive organ and the female reproductive organ. So here we can induce the flowers to undergo self-pollination as well as cross-pollination. In reproduction chapter already we have studied about self-pollination and cross-pollination. So cross-pollination is the one which is taking place between two different plants but belonging to the same species. So that will definitely bring out the variation. Self-pollination is the one which is taking place within a flower or between two flowers of the same plant. So there you can't expect much variation to take place. So here this plant exhibits different physical characters, the two contrasting characters. In this garden pea plant we have several contrasting characters present, which is easy for Mendel to experiment it out. And this particular plant have very shorter lifespan. So within a short lifespan of time or within a short duration of time, we can able to see the progenies. So here, always when you take research work, repetition of the results is very, very important. So this particular advantage, that is the shorter lifespan of the plant, made Mendel to redo his experiment and proofread his readings several times. Apart from this plant is easily available. We can able to easily grow them in the natural condition under our garden. So these were the reasons behind the selection of this plant as the experimental material. So apart from that, what else what are the 7 different characters they have? So here if you see, Mendel observed 7 traits that are easily recognizable and apparently only occur in the 2 forms. So if you take 2 different pea plants, the one flower will have one character, the other flower will have exactly the contrasting character for that. So this was the main reason behind choosing this plant for the experiment. So here the first one, if you take flower color. So in this garden pea plant we have 2 different flower colors, violet or purple and then the white. This purple is the dominant color and white is the recessive color. Then we have seed color, yellow and green. Then we have flower positions. The position of the flower on the branches, it is again 2 types, axial and terminal. And then we have the pod shape and then we have stem length, pod color and then seed shape. Let us see it one by one. So if you take out the 7 characters, the first one, it is completely required to memorize this character, 7 characters, which one is dominant, which one is recessive. Based on this only case-based question, numerical questions will come from this chapter. So here if you take the first one, the seed shape. So here we have 2 different seed shape. One is the dominant shape called as the round. The shape round is said to be the dominant and the opposite one is wrinkle. So if you look at the surface of the garden pea seed, if the surface is smooth, it is called as round. If the surface is having some shrinkage, it is called as the wrinkle. So seed shape, we have 2 types, round seeded and then wrinkle seeded, in which round is dominant, wrinkled is recessive. Then if you see the seed color if you take, yellow is the dominant and green is the recessive. So yellow color seed is called as dominant and green color is called as recessive in nature. So then if you take the pod color, pod is nothing but the outermost covering of the seeds. It's called as what? Pod. So here if you take, green is the dominant one and yellow color pod is called as the recessive one. So we have pod color, green and the yellow. And then we have the pod shape. If you look at the pod shape, we have the dominant character called as inflated. Inflated in the sense, the pod structure, it remains to be smooth. You don't have any constrictions or ups and downs. The surface of the pod will be very smooth without any constrictions. That is called as inflated. The opposite recessive one is constricted. Constricted in the sense, see, you can able to see the shape of the presence of the seed from the outer side of the pod itself. So you will have some constrictions over there. Some ups and downs or the furrows will be there. That is called as the constricted. So inflated means it is dominant. Constricted is nothing but recessive. In flower color, if you take, violet color is the dominant and white color flower is the recessive one. Then flower position. If you look at the flowers where it is attached over the branches. Here we have axial. Axial means all over the branches. All over the branches you can able to see the location of the flower. The opposite one is terminal, the recessive character. Terminal means end, towards the end of the branch or towards the end region only you can able to see the flowers present over there. And the last one is stem height. Here some plants are having the tall nature, some plants have the dwarf nature. That means the taller plants have a gene which is responsible for their tallness. More amount of growth hormones will be secreted, so they will be taller in nature. But if you take the dwarf plant, it always remains to be short. So that is called as the recessive character. And altogether, Mendel worked on this particular plant material called as Pisum sativum. I told you all the reasons. What is the reasons which is responsible for selecting this particular plant material as the experimental property, and of course that question, why Mendel has chosen the garden pea for his experiment, is often repeated question in your board exams. And based on this experiments with this particular plant species, Mendel has proposed 3 laws. The first one is law of dominance, then we have law of segregation, and then we have law of independent assortment. These are the 3 laws of inheritance proposed by Mendel on working out the monohybrid and the dihybrid cross experiment. So far in this video we have discussed about the basic terms which are related to genetics, and then we have discussed about the experimental materials and the reason behind the choosing the experimental material by Mendel. So the next session we will be dealing with the experiments which are related to this Mendel's experiment. Thank you children, have a nice day. Go through the sessions continuously so that you can be updated with the informations here. Thank you.